Okay, today we're going to start the process of protein synthesis. And what you need to understand is that protein synthesis is a two-step process, and the first part is transcription. Okay? Now, you can remember this, that it's called transcription, because what we're doing is we're copying one gene from DNA into an RNA molecule. And when you ask for a transcript of something, that's basically what you're doing, right? You're taking something that's spoken and you're putting it into written form, but it's in the same language, right? So if if you write away to a show and want a transcript of a show on TV, what's going to come back is, instead of a videotape of the show with all the spoken word, what's going to come back is a script, basically. Okay, so transcription is making a copy of one gene from DNA into an RNA molecule, but it's still all the same language. It's still nucleic acid language. The reason we do this, remember, is because DNA can't leave the nucleus. Now remember, DNA's information is supposed to get out of the nucleus to the ribosome, right? Because that's where protein synthesis occurs, and you remember that from units 2 and 3. But since DNA can't go itself, it's got to send these little RNA molecules out to do the work for it. Now there are three types of RNA that are going to come into play with protein synthesis. The first type is mRNA, and the M stands for messenger. And this is the RNA that we normally think of when we make a transcript like this, when we do transcription. This messenger RNA is going to carry the gene that we need to the ribosome. It's going to be the code that we're going to actually read to build the protein. So that's messenger RNA, and it kind of looks like, if you see, this is a picture of it right under here, it looks like the RNA molecule that you all built in class. The second type of RNA that's going to be important here is tRNA, and the T stands for transfer. And the transfer RNA is important because it bends into this weird, funny shape. And what happens is you end up with one active area here, right here at this end. That's the active area, and we'll get into that in the next lesson when we get into translation, the second step of protein synthesis. But at the other end, it's got a docking site right here for amino acids. So the tRNA's job is to carry amino acids. Amino acids bind to that docking site where the arrow is, and the tRNA will carry amino acids into the ribosome. Now remember, we're building proteins, right? So proteins require amino acids because amino acids are the building blocks of proteins. The third type of RNA that's going to be involved here is rRNA, which stands for ribosomal. Ribosomal RNA. Now, this picture that you're seeing is a whole ribosome, where part of that is protein. A ribosome is made up of protein, but it's also made up of this rRNA, or ribosomal RNA. So these are three types of RNA. All of them are made by transcription, okay, but they all kind of do different things. Messenger RNA is going to stay like you see it. tRNA is going to fold up into this funny shape and start picking up amino acids, and rRNA is going to leave and become incorporated in ribosomes when they're synthesized. Alright, so now we've got the process of transcription to discuss, and the first thing I just want to draw a distinction for you here is this is going to look a lot like replication, and it's going to function a lot like replication. But what I need you to remember is, from the first lesson, the one on replication, that replication is a very different process because it's not part of how DNA normally functions. DNA's normal function is what you're seeing here in front of you, transcription, and then eventually translation. The only reason that replication exists is for cell division. Okay, so that has nothing to do with this process that we're about to go through, even though it's going to look very, very similar. It's going to start here with a DNA molecule just like before, and in the next step, the molecule is going to start to unzip. Now, as it unzips, new nucleotides are going to be filled in, and this time we're doing RNA nucleotides, so we're going to use U's instead of T's. So any place that you see an A, it's going to bond to a U. All right, and we keep going, right? Okay, so this is going to fill in, and notice, as you're doing this, we're not copying the second side of the molecule. This side of the molecule is not getting copied. RNA is single-stranded, right? So we only have to copy one side of the molecule to get an RNA molecule. Okay, so we're copying, and what we're going to end up with is something that looks like this. Okay, now notice we also did not unzip the whole thing. To get this piece of messenger RNA that we've made, we only unzipped it to there, right? Because again, RNA is a shorter molecule. It only contains one gene. Notice also at the end that the DNA that we have here snaps back together once it's done. Okay, so another difference between this and replication. Now, at the end of transcription, this is our RNA molecule, and it's going to go off to the ribosome. So this is an mRNA that we've just made, and it's going to leave the nucleus at this point and go to the ribosome. This is a gene that it contains, and that gene is going to direct the synthesis of some protein. So this is the code for how you put together a particular protein. So that's what the messenger RNA is going to do. All right, so we've copied one side, DNA snaps back together, the RNA leaves the nucleus, snakes its way out, and goes to the ribosome for protein synthesis, the second half of protein synthesis. Don't forget also that, like replication, transcription is controlled by enzymes in the nucleus, right? Any process dealing with a DNA molecule has to take place in the nucleus because the DNA can only exist in the nucleus, right? It can't leave the nucleus. So this is being controlled by enzymes, and it is happening in the nucleus. That's pretty much it for step one. That's transcription, okay? Now, what you should do with this is go back to the Venn diagram from the first homework. Look at the second Venn diagram where you are comparing and contrasting replication and transcription, and you should be, now that you have this information, filling that out.